Good afternoon. It is my pleasure to introduce Professor Tom Elliott, consulting microbiologist and deputy director of the Medical University Hospital Birmingham National Health System Trust, and also a pioneer in clinical demonstration that copper alloy surfaces reduce bacteria. Professor Elliott, the floor is yours.
Good afternoon, ladies and gentlemen. I'd first of all like to thank the organisers for giving me the opportunity of speaking to you today regarding a clinical trial we've been carrying out in Birmingham with the use of copper. If I could have the first slide, please. So what I'm going to be talking about is the use of copper for the prevention of microbial contamination of the clinical environment. Next slide, please. Thank you. My presentation is going to take 5 components. First of all, I'm going to review very briefly for you the problem we had in the UK, as in many other countries, the problem associated with healthcare associated infections. Secondly, I'm going to review briefly for you some of the previous observations regarding the antimicrobial activity of copper, and then I'm going to be looking at some laboratory studies which we've carried out in Birmingham in the UK, and then to describe for you in some detail the clinical trial which we have carried out on one of our medical wards at University Hospital, and to finally give you some conclusions bringing it all together. If I could have the next slide, please. In the UK, we have approximately, for every 9 patients who come into our hospitals, we have approximately 1 of those patients who will develop a healthcare associated infection. And that means and equates to 300,000 infections per annum each year. And that causes over 5,000 deaths. And this is national data which has been published within the literature. And the cost to our National Health Service is over £1 billion per year. Now, when we're considering the prevention of these infections, we need to consider the source of the microorganisms. Where do they come from? And I was able to listen to some of your earlier discussion this morning, and it's very evident that we are all aware that the environment is a potential contaminant, is a potential source for these organisms which can cause infections in our patients. So not only is it the patient's organisms themselves, their endogenous organisms, but also the exogenous source from a doctor, from other healthcare workers, but also the environment, which is important. And this is why I'm very excited today to present to you our results relating to the use of copper to see whether or not that can reduce down environmental contamination. If I may have the next slide, please. If we consider briefly the observations made previously about the use of copper, it's been very evident for many, many hundreds, if not thousands of years, that copper has antimicrobial activity. When the earliest books published, the Smith's Papyrus, which described the use of copper in drinking vessels, and this resulted in a reduction in people who drank the water getting subsequent infections. We've seen the use of copper on the bottom of boats to prevent fouling with bacteria and other organisms. And of course, in the agricultural field, for example with grapes, we've seen the use of various copper sprays, copper sulfate for example, to kill fungi. So we're very aware that copper has antimicrobial activity. What we have been slow at doing is harnessing this property to see whether in the clinical environment it offers advantages to us. If I may have the next slide, please. Thank you. So in summary then, we have seen in the 19th century an array of different descriptions, albeit not in proper clinical trials, but we've seen descriptions of immunity to cholera and treatment of arthritis. There's been descriptions in the 20th century of miners working in the copper industry not getting arthritis. More recently, we've seen studies looking at copper in inflammatory conditions, and one would predict that in a clinical environment we're going to be seeing much more now in terms of the use of copper and its application to these inflammatory conditions, asking the question, can we use copper not only for its antimicrobial activity, but also for its anti-inflammatory activity? And one final comment on that slide, you'll see the relationship to copper and Legionella, particularly in water systems. Has copper in fact protected our water systems from Legionella? And it's an issue which we need to explore in more detail. So in summary then, we've seen observations over many years regarding the use of copper. What about its use in a clinical environment? Next slide, please. So moving on to the antimicrobial laboratory studies, and I know that Professor Keevil is going to outline this in a more detail for you, but Professor Keevil, an expert in the activity of copper against organisms, has carried out detailed in vitro investigations and has widely published on this. And he will outline, I hope, to us later this morning that he has demonstrated quite clearly that copper kills a variety of microorganisms, including the methicillin-resistant Staphylococcus aureus and Escherichia coli. And this is exciting in terms of asking a question now, can the copper be used in a clinical environment? We've also confirmed Professor Keevil's outstanding results in our own laboratories, where we have obtained fresh clinical isolates from patients and asked the question, if we test these organisms and challenge copper with these different organisms, will the copper deal with them? And on that slide, you can see there is a, sorry, if you bring the next slide, thank you. On this slide, you should see that there is an array of clinical isolates which we have looked at. Escherichia coli, methicillin-sensitive Staph aureus, the intermediate strains of MRSA, the epidemic strains of MRSA, 15 and 16, which we commonly see in the UK, Acinetobacter baumannii, Candida albicans, Klebsiella pneumoniae, and Enterococcus faecium. And what we have done with these organisms, we put a large number, 10 to the 7 coliforming units on pure copper and compared it to stainless steel. We incubated the stainless steel and the copper then for 3 hours and looked at the numbers of organisms present on those two metals after the 3 hours had elapsed. Next slide, please. And this gives you a summary of the results. On the left-hand side of the slide, you'll see the microorganisms, the ones I've just described. You'll then see the comparison between the numbers of organisms which we have seen in terms of log reduction. On the right, the stainless steel, you'll see that the organisms remained viable. There was no reduction whatsoever on the stainless steel for all those organisms. In comparison, in the middle, the pure copper resulted in a significant reduction of all those organisms on copper surfaces. And there was a large log reduction for both gram-positive, the Staph aureus, gram-negative organisms, for example, E. coli, and also for the fungi Candida albicans. So in our own laboratories, we demonstrated and confirmed Professor Keevil's earlier findings that a whole array of organisms can be killed when they come into contact with copper. Next slide, please. So the summary of the laboratory studies to date, we have demonstrated and found that copper is inherently antimicrobial, and it's what I would call an intelligent metal. This is something which is helping us, and it's killing organisms all the time. It inactivates bacteria and fungi, and also in the literature, viruses as well. And we have confirmed that it kills the clinical isolates. But the question is, will it work in a clinical environment? Next slide, please. The possible mechanisms of copper, and I think that Professor Keevil perhaps will go into this in more detail, are still unclear. So for those of you who are wondering, well, copper is killing it, but how is it working? Well, this is two possible mechanisms. It may be acting on different enzymes, which can damage the metalloenzymes in particular, or it may be affecting the regulation response of the organisms to stress. Whatever the mechanism is, and I think further work needs to be done on this, it is very evident that it kills a whole array of bacteria and viruses and fungi, and this has the potential, as I said earlier, for an exciting application in a clinical environment. Next slide, please. I now come on to the clinical study which we carried out at University Hospital Birmingham NHS Foundation Trust. Next slide, please. Our hospital is a 1,200-bed hospital on two sites. And what we did was we identified on a busy medical ward areas which we felt was ideal to substitute our usual product for products made of copper. And we had various products produced for us, and I'm most grateful to the CDA for organising this for us. But you'll see some of the products on this slide, and it ranged from light switches, which had copper plates on the actual switch itself. The door handle is made of copper. The one in the middle is a push plate on the entrance to a busy medical ward. The next one is just a light switch on its own. And the final picture on the right-hand side is a toilet seat, both the lid and the base, as well as other items, including taps and grab rails in the bathroom. We had an opportunity to explore and ask the question, would these reduce down contamination in a busy medical ward? Next slide, please. The next slide shows you an outline of our medical ward we selected. And we deliberately challenged copper in a busy medical ward, which has 28 beds. The ward is very busy in terms of the throughput of patients who are commonly only in for about 4 days. We have a main ward. On your right, you'll see the entrance to the ward, to the main door, which had the push plates on. On the right-hand side and left-hand side, there were bathrooms, isolation rooms, kitchens, and then the main ward towards the other end were bathrooms as well. And what we decided to study following a preliminary analysis was to look at, in particular, the toilet seats, where we found a lot of contamination, to look at the taps and also the push plates going into the ward. We designed the study, next slide please. Thank you very much. We designed a study, as I said, on this busy medical wards. We put 3 products in, zinc taps, which contained 60% copper, copper zinc, push plates, 70% copper, a toilet seat, which was a copper composite, approximately 70% copper. And we have shown, as Professor Keevil's also shown, that copper of this concentration, this percentage, will also kill organisms in the laboratory. And what we did was we compared it to our standard items, the controls. So in other words, for example, the push plates, these were aluminium, the toilet seats were the normal plastic components. We did weekly sampling, swabbing these items over a 10-week period on 2 time points. First thing in the morning, 7 a.m, and towards the end of the day after a busy clinical period at 5 p.m. The study took place over 10 weeks, and after we were halfway through the study, all the items were changed round with control items to overcome user bias. So in other words, for example, we had some toilets which had the copper toilet seats, others had standard. And after 5 weeks, we changed those over so to overcome user bias. And the end point was the numbers of microorganisms on the surfaces. Did the copper work? Next slide, please. So these are the 3 items which we studied so far. We've got plans to study other items in a further clinical trial. So we looked at push plates on your left, the toilet seats, which, and I'm sorry it is just before lunch for you to talk about toilet seats, but the patients were very interested with these seats and wondered whether they were being cleaned appropriately. But once they got used to them and we explained the study, they were more than welcome to use them. Similarly, the taps you can see on the right hand side, the standard taps were made with the copper within it. We also implemented on the board an increase in cleaning, enhanced cleaning, to make sure that we weren't overburdening our control surfaces and our copper surfaces with organisms. And to ask the question and to be quite clear that we weren't getting contamination because we weren't cleaning appropriately. So we were cleaning all these items every 2 hours during the working day. Did copper make a difference? Next slide, please. Now, what I've done for you today is I've given you the results for the 3 items we've looked at, the toilet seats, taps, and the push plates, on different slides. This slide shows you the results of the toilet seats. On the left-hand side is a sample location and time, and we sample both the top and the underside of the toilet seats in the morning and later on in the day. The next column shows you the median number of microorganisms on the control items, the plastic toilet seats, compared to the copper. And if we just take you through the top line, for example, you can clearly see that on the top of the toilet seats, 2,190 organisms, colony-forming units, were found on the median number over those 10-week periods. And this was despite enhanced regular cleaning every 2 hours. However, if you look at the numbers of organisms on the copper toilet seats, you can see for all those sample points a significant reduction. So 2,190 versus just 5 on the copper toilet seats. And this was a defined area we sampled. It was strictly controlled so that we would know that we had sampled comparable surface areas and comparable areas of the item. Similarly, for the underside of the toilet seats, again, interestingly, there are fewer organisms for obvious reasons on the underside of the toilet seats, but again, you can see there was a significant reduction. And if you look at the p-values on the right-hand side, you can see that for all those samples over a 10-week period, there was a significant reduction with the copper toilet seats compared to the standard plastic. Next slide, please. If we move on to the door push plates, again sampled 7 a.m. in the morning, 5 p.m. in the afternoon. Fewer organisms on the push plates compared to toilet seats, which one would predict. But again, you can see that the numbers of organisms on the copper were greatly reduced. And again, for both these time points, 7 a.m. and 5 p.m., when we compared the copper item to the standard aluminium push plates, we reached a highly significant difference between the two. The copper was killing the organisms. Next slide, please. Finally, if we look at these zinc taps, And here we sampled, you'll see on the left-hand side, both the hot and cold taps, and we sampled the areas we felt were likely to be most contaminated with hands touching the taps. Again, sampled, as I said, at 7 a.m. and 5 p.m. You can see the medium number of microbes in the control. These were taps which were our standard NHS taps, which were chrome plated, organisms present on all of the taps. And you can see the range was quite dramatic, ranging from 0 to 5,040 organisms. You hope when you go to a sink to wash your hands, you'll end up with clean hands. With the chrome taps, I would guess, unless you were lucky, you may end up with more organisms on than you started with. And again, you will see that for 3 of the sample points, we reached significance in terms of a reduction in the numbers of organisms. Next slide, please. All copper items which we had sampled harbored 90 to 100% fewer microorganisms than their control equivalents at both 7 o'clock in the morning and 5 p.m. and that this reached statistical significance using paired and non-paired, non-parametric statistical analysis for 9 out of the 10 areas sampled. So I was going to finalize just to say that these results demonstrate quite clearly that copper offers an opportunity to significantly reduce the numbers of common clinical microorganisms, not only in the laboratory, but in a busy clinical environment where you will have a lot of debris, protein, serum, etc., even fecalant material. The copper still works, which when we found these, we were absolutely excited by it, because it means that here we have an intelligent metal, a metal which retains its activity. Many of these items had been on our ward for over 6 months already. Some were tarnished, some were not. The copper still retained its activity. So I would conclude that the use of copper in combination with optimal infection prevention strategies may reduce the risk of patients acquiring infections in hospital and other healthcare environments. I'd ask you this question: if copper is able to offer this for us, shouldn't we be using items which are made of copper? Finally, if I could have the final slide, please. Next slide. I'd like to thank various people, Dr. Casey and Professor Lambert at Aston University, and Lisa Maryshenko, our senior nurse, who helped us with the study. The Copper Development Association have been fantastic, who gave us an educational grant. Angela Vesey and Mark Turr particularly, and all the suppliers of copper-containing products who have been most generous in terms of making the devices and items for us to allow us to carry out this clinical study. And finally, the estates team at our hospital, who have been very compliant in putting these items in. Thank you all for allowing us to carry out this study, and thank you all for your attention.
I will proceed now. My talk is about the regulatory testing we had to do in order to achieve registration in the United States. The testing was a short part. It took about a year and cost well over $1 million. The hard part was the total of 4 years negotiation with the EPA about what were the tests, how would they be conducted, what would be acceptable from them, and to negotiate the claims. And this is the first time they ever allowed a solid material or a coating to get a public health claim in the United States. Up until this point in time, only gases and liquids, which are sanitizers and disinfectants, were allowed to make the claims we can make at this point in time. Let me begin. Mother Nature has been very good to copper. It's an element from the periodic table. We know its weight. We know it's a solid at room temperature. It has an interesting, unique color, which I won't explain why, but there's a good reason that a physicist could explain. It also has a lot of interesting properties that have made it useful for man, not only in antiquity, but to this day. It's inside your cell phone. It's inside your computer. It is truly an interesting material. It is highly ductile and malleable. That means you can shape it readily. It has very high thermal and electrical conductivity, and it has good corrosion resistance, and it's highly recyclable, but it has a unique property that we have not exploited until this point in time. It is antimicrobial. And several people talked about ancient societies utilizing copper. I will not dwell upon these things, but we lost this knowledge as time went on. Before they did all of this, before there was any germ theory, so it's really quite amazing. There was one study done about 3 years ago, maybe it's 4 years ago, by Professor Reed, who was then at the University of Northumbria. He worked in the laboratory, but he also worked in the field in the Punjab in India, and he demonstrated that brass vessels filled with water overnight, that the E. coli were eliminated. And it's just another indication. And the problem with the society in India at present is they're using plastic vessels because they're cheap and readily available. They become ill, they can't work, they can't feed their families, so it's a serious problem. And it's just an indicator. It shows that copper can work in many different environments, not only on touch surfaces, but also in aqueous environments. How did I get this idea? I came upon a nurse's student study conducted in 1983. It was a little project and it was written up in a newsletter. It really wasn't even a technical paper. But basically, they found heavy contamination on stainless steel and aluminum doorknobs, but very little bacterial growth on brass. And then they went back and they purposely contaminated them, and they found it happened again. The brass would knock down the bacterial load and nothing would happen on the stainless steel. So on the basis of that, I started working with Bill Keevil, but I wanted just to give you some statistics that people are interpreting in different ways. Hospital-acquired infections are a serious problem in the United States. 2 million people are infected each year. 100,000 die each year from these nosocomial infections. And the cost, you've seen numbers being thrown around. It's probably 20 billion is probably conservative. The last official number is about 15 years old. That was 5 billion. It's probably closer to 30. I think the number of infections are down because the hospitals are not reporting all of them. If someone dies, they'll say, well, they died of heart failure, but the infection could have been a significant contributing factor. So it is a serious problem. And in spite of all of the hand washing and the various alcohol gels, etc., we still have this problem. And it's a difficult problem, and we believe that copper is part of the solution. We don't think it's the solution. There is no one way of solving this problem. But you can look in the newspaper and you hear about it all the time. It's jumped to the general population. It's in schools. Students are catching it. It's actually in sports figures. It's turning out, or football players are having MRSA infections. And it is generally in society, and it goes from the nursing home to the general community back to the hospital, and we get this vicious cycle, and we have to break this cycle. This is data which Bill will describe more thoroughly, I suspect. But this is our first data on MRSA, and basically we showed that pure copper killed. We had a 7-log drop in 90 minutes on MRSA. It took a little bit longer on a brass, the C240, and even on an alloy that's outside the range we submitted to the EPA, a 55% copper alloy. We ended up, if you do it, look, this is a semi-log plot, you've got a 2 or 3 log drop. That's still significant. And stainless steel, we get really very little reduction, and the reduction we get, we believe, is due to evaporation. Stainless steel is our experimental control, and an EPA test, which I will describe, it is their mandated control surface. These are some of the organisms of interest. We have a little bit of data on some of them. We have a lot of data on several of them. The light blue ones, the E. coli 0157H7, the methicillin-resistant Staph aureus, regular Staph aureus, Enterobacter, and Pseudomonas are organisms that the EPA mandated that we test. And those were the 5 organisms, and I'll repeat that information later on. And we have various bits of data on all of these organisms. There is something in the United States called FIFRA. Now, it's a mouthful. If you want to make an antimicrobial claim in the United States, it falls under the jurisdiction of the EPA. I didn't realize that when I started this. Maybe I wouldn't have even done it if I knew I had to get involved with the EPA, but it falls under their jurisdiction. Specifically, it falls under the jurisdiction of the Federal Insecticide, Fungicide, and Rodenticide Act. That's what FIFRA stands for, because in the minds of the EPA, a bacteria is a pest, and FIFRA is for controlling pests. All pesticides must be registered, properly labeled, demonstrate they present no harm to the environment. But you only have to demonstrate they work if it relates to human health. If you have something you want to spray on a crop, they really don't care if it works or not. They said the farmer will figure this out. But for human health, they have the obligation to demonstrate it works to their satisfaction. So we conducted tests under GLP, Good Laboratory Practices conditions, at an EPA-certified laboratory. We were not in control of the data. The EPA audits this laboratory. All of the data is logged into books. It's checked. All of the equipment is calibrated on a regular basis. The strains are certified. It's a very, very rigid and expensive process. And we conducted these tests in one laboratory in Minnesota. And we looked at 3 test protocols, which EPA approved. We modified 2 and created a new 3rd one with them, which I'll describe. We looked at 5 copper alloys down to 65% copper, and we conducted these tests. And I won't go into the details. I'll skip over. We followed general microbiological procedures, and basically, we measured the amount of bacteria on the surfaces at the end. These are the certification of which strain we looked at, and we got some very interesting results. We had to use 3 different manufactured lots of material. There were 3,000 specimens involved in these tests. And of course, these are the 5 bacteria. And it's very interesting. We have MRSA, methicillin-resistant staph, and staph. They wanted both of them. And there's an interesting twist in the data when I show you the final summary, which I'll comment on. Just to give you an idea what these alloys are, C110 is copper, like copper on a roof. C260 is yellow brass. Common yellow brass is called cartridge brass. C510 is a phosphor tin bronze, a very common alloy. Copper nickel, 9010 copper nickel, and a nickel silver, which looks silvery, but it isn't silver. It contains copper, nickel, and zinc. And I'll show you the first protocol. But the claim that goes with it is we kill 99.9% of the bacteria, those 5 bacteria, the only ones we tested, within 2 hours. And here's a bar chart. And this is just an example. You just plot the data and show it on a table. They don't plot data. But the blue is the initial concentration on the copper and the stainless steel. Notice the stainless steel drops about 1.5 logs or 2 logs. That's due to evaporation. But look what happens on the copper. That's on Staph aureus on the left, and on the right you have the Enterobacter. And all the data will show the same. The second test is basically a wear test. We're going to rub the surface back and forth and conduct a test afterwards to see if it still works. They want to know, does the antimicrobial properties go away? Are they worn out? And the short answer is no. A doorknob doesn't wear out. typically. And this is the schematic of the result. You start out, you measure the testing. On the left, we're just measuring efficacy. That we inoculate the surface, we go through 6 wet and dry cycles of rubbing it with a cloth. And I'll show you the device that rubs it. It's not a human doing it, it's a device. And then we measure efficacy in the end. And if the material still, if it looks the same, it didn't wear out. They wanted this test done, I suspect, because they've tested other coatings that contain silver ions and they didn't pass this test, so they wanted us to see if we could pass this test. This is the device, this copper sample. You can see it on the left. And this weight, which is on the right, I turned it over, pushes down on a cloth with a certain foam, and it gets pulled over the surface back and forth, and then you spray it with water so many centimeters away with a nozzle so big, and then it gets run across the thing again. Humans don't touch it. The machine takes care of all of the movement, so it's reproducible. And they look identical before and after. So we know we haven't destroyed the antimicrobial properties of copper by going through simulating cleaning it. And the third one is the most difficult test to pass. And basically, they made us inoculate samples every 3 hours. And over a 24-hour period, we had 8 inoculations. And again, I said they just plot data and you show it on a table, but I'd like to graphically show you what it means. The upper line that keeps on increasing is the stainless steel, the EPA mandated control. The sawtooth curves are what's happening when you keep on putting on the bacteria. Notice we start to get some survivors towards the end. There are lots of dead bacteria underneath. The copper is still diffusing through, and it just doesn't get all of them. If we waited another hour or so, it would probably knock it down further. But just to make sure, we took these bacteria that survived and we ran through the normal 2-hour test and they were killed. They're not developing any resistance to copper. It's highly unlikely. This is it as a linear plot, which I think is much more revealing. Notice the increase in bacteria on the stainless steel and look at the background number on the copper. So it's quite dramatic when you look at it linearly versus on the semi-logarithmic plot. And this is a summary of our data. We have 180 tests here, 5 different bacteria, 5 alloys, 3 test protocols. In all but 6 of these tests, I have greater than 99.9% reduction. And in the other 6, we have between 99.3 and 99.9% reduction. So for this last test, we couldn't claim greater than 99%. This is the continuous reduction. We claimed a 99% reduction. But notice there are more survivors, there are a little bit of survivors on the Staph aureus versus the MRSA. MRSA's been around 30 years. How long has Staph aureus been around? Eons. This has evolved in the outside world and went into the hospital. The MRSA evolved in the hospital and came out and back again. And it is more sensitive to copper because there wasn't that much copper in the hospital versus in the outside world. These organisms are really more robust in many ways. They've been exposed to many things over their evolutionary development. This is the legal document issued on February 29th of this year, and it actually covers 275 alloys. This was a mathematical error we made. They made us remove all alloys that contain 0.1 or greater of 3 alloying elements: lead, arsenic, and chromium. And I can tell you why another time. I won't take our time. But basically, these are the claims we're allowed to make. Antimicrobial copper alloys continually reduce bacterial contamination, achieving a 99.9% reduction within 2 hours. They kill greater than 99.9% of gram-negative or gram-positive bacteria within 2 hours. They deliver continuous and ongoing antibacterial action, remaining effective in killing greater than 99% of bacteria within 2 hours, even after repeated wet and dry abrasion and recontamination. And when cleaned regularly, antimicrobial copper alloy surfaces kill 99.9% of bacteria within 2 hours and continue to kill more than 99% after repeated contamination. You get the idea. These were legally negotiated claims, and the data supported these claims, and they gave us the right to make these claims. This will allow us to promote copper alloys, this unique attribute, intrinsic property of copper alloys. Now, they made us make certain statements, and they make a lot of sense. The use of copper alloys is a supplement to and not a substitute for good infectious control practices. Users must continue to follow all current infection control procedures related to cleaning and disinfecting the environments. There's also a statement in there about cross-contamination. They made us put it in because we didn't test for cross-contamination. The budget wouldn't allow it. If we go back and test for that, they will allow us. But basically, we're not advocating that it's a substitute for good hygienic practices. We're saying it's one of the tools you can use to reduce, hopefully reduce infections. And we've continued, Bill continues to put out very interesting peer-reviewed journal articles on various organisms, and many of them are not covered by our registration, but they are peer-reviewed articles, including viruses. There's one on C. diff. We actually kill C. diff spores. And now we're conducting our trials at 3 hospitals in the United States with Department of Defense money. And hopefully that will give us the real information we need, a reduction in infection rates. And in conclusion, copper alloys are naturally antimicrobial. They provide lifetime efficacy and durability versus coatings. They are homogeneous and solid. The antimicrobial property will not wear off. It will not scrape off. They are superior to any surface or coating additive, and are the only commercially solid material with EPA FIFRA public health registration in the United States, and we are allowed to legally make these antimicrobial claims. And my final thought is copper alloys should be utilized in those applications where the unique intrinsic antimicrobial properties will benefit human health. Thank you. It's my pleasure to introduce Bill Keevil. Bill is a professor of microbiology at the University of Southampton, and we work very, very closely together. And I hope he's enjoyed the interaction half as much as I've enjoyed interacting with him. Thank you, Bill.
Good afternoon. It's a pleasure to be here, and thank you for the invitation. As Harold said, it's been a pleasure working with him and a whole range of colleagues in the copper industry. I describe myself as a simple microbiologist. I'm delighted to work with metallurgists and engineers who can explain to me some of the physical and chemical properties of copper and how they could be usefully used in the built environment. So I'll take you through some of our research, which will hopefully convince you of that. Now, the title of the talk is a very dry one in terms of laboratory studies, but really what I should call this talk is stealing from a Hollywood film, Back to the Future. And the reason for that, as you've already heard from some of the previous speakers, is that antiquity tells us a lot about the beneficial properties of copper. We're slowly rediscovering those, and it's given us insights in how we should be moving forward to the future. So I think Hollywood got it right. Back to the Future, I think, is the success story of copper and will be for the future. I should also acknowledge at this stage my collaborators, Sarah Warnes and Louise Weaver in my laboratory, and Harold Michels himself at the CDA. It's been a very fruitful collaboration. Okay. You've already seen this slide in various guises previously, describing how copper has been well recognized in antiquity. It's worth reminding you actually about what happened in France in 1850. Paris went through 3 waves of cholera in quite severe epidemics, and thousands of people died. Now, even before Pasteur recognized and published the germ theory, it was recognized that copper workers were immune to cholera infection during these very severe cholera epidemics. So already there was a handle, even in France in the 1800s, that there was some health benefit being conferred by copper to such a dangerous disease such as cholera. Harold has also mentioned the more recent studies of Dr. Phyllis Kuhn in Philadelphia looking at the door handles. And it's rather surprising that no one actually picked up on her published study until we were asked by the Copper Development Association to get involved looking at the antimicrobial properties of copper. So I'm delighted to recognize what Phyllis Kuhn first recognized, and I'm more than willing to publicize what she first proposed and warned the medical fraternity that by switching from copper to other materials would increase health risks in hospital. We should acknowledge that. It's been around over 25 years. Okay, now within our research, which has been funded by the International Copper Association in collaboration with the Copper Development Association, we have been looking, for example, at waterborne pathogens, particularly Legionella pneumophila and E. coli 0157, and we've shown how copper tubing will reduce the numbers of these pathogens in drinking water systems. We then moved on to our dry surface work, initially looking at foodborne pathogens. Again, E. coli 0157, because it's also known as the burger bug and has been responsible for many outbreaks of foodborne disease. And also Listeria monocytogenes, the pate bug, as some people might call it. And this was a pathogen that we thought was going to go away 10 years ago. It's come back in even greater numbers in the USA, Europe, and around the world. And I'm pleased to say that I haven't got the time to show you the results today, that copper and copper alloys were able to successfully kill both E. coli 157 and Listeria. What I want to concentrate on today is the work we've done on the hospital-acquired pathogens, and I'll be showing you some of the data we've developed with MRSA, VRE, Enterococcus, C. difficile, and Acinetobacter baumannii. I'll also show you some brief data on the virus work we've done with influenza A and adenovirus, and some very recent work which we've just sent for publication on fungi, Aspergillus, but also Candida, Fusarium, and Cladosporium. Harold has already described some of the alloys that we've investigated. We've looked at quite a range of alloys supplied by the copper industry, traditional coppers, brasses, bronzes, copper nickels, and copper nickel zinc. And of course, we've wanted to compare these against competitor surfaces, specifically stainless steel, but also looking at various plastics and polyethylenes, which are also used in the built environment. We use quite a simple test where we grow the pathogens and then we inoculate them at different concentrations onto small coupons of each of the test materials. We incubate at different humidities for different amounts of time, and then we recover the pathogens from those surfaces and we attempt to culture them. And we also take them through other biochemical tests to reassure ourselves whether they are truly alive or dead. And to do this, we've developed in situ microscopy methods. We're fortunate to have a patented microscope system which allows us to look at a whole range of different surfaces. So, for example, we could scan taps from a hospital if necessary, and we hope in our collaboration to be able to do that in the future. Now, you've already heard about how, regrettably, hospitals are very dangerous places. And there's this very serious risk of transmission through contaminated hands. And this is just a simple cartoon to say it's really impractical to keep everyone's hands clean without going through some cleaning, decontamination procedure on a regular basis. You've already heard a lot about hospital-acquired infection and pathophysiology. It's well recognized some of the risks involved. There's invasive procedures during the surgery and treatment procedures. There's inappropriate use of the antibiotics. There's organizational risks such as contaminated air conditioning systems, particularly for Legionella, but also we're more aware now of fungi being spread through HVAC systems. There's the staff themselves and visitors coming into the hospital. How can we keep those clean? That's an incredibly difficult task. And then, of course, there's the patients themselves who have, due to the severity of their illness, their age, their immunocompromised state, and inevitably how long they're going to stay in the hospital, these are all increasing risk factors. You've already seen much of this data, so I'm not going to repeat it. Hospital-acquired infections are a very serious problem. They cause high morbidity and mortality, and they cost an awful lot of money to treat. Again, you've heard a lot already about MRSA, originally described as methicillin-resistant Staphylococcus aureus, but in reality now called multiple antibiotic resistant. And it's getting more and more difficult to treat, and when patients acquire this infection, if clinicians are fortunate, they can cure the patients. In many cases, the morbidity is very high, and certainly it's resistant. What this means is we're losing, if not already lost the fight, to control VRSA infections. What that means is that prevention must be better than cure. Now, when we actually look at some of the published data for MRSA around Europe, I'm embarrassed to say that the UK is in red, showing that we have high rates of MRSA infection. You'll also notice that Spain, Portugal, Italy, and also, it has to be said, Greece, are also reporting very high rates of MRSA infection. We're struggling to hold on to this infection. Now, when we look at how the built environment is constructed, and particularly in healthcare facilities, you will notice there is a lot of stainless steel. Why? Because it looks good, it's corrosion resistance, and a lot of people think mistakenly that it's an aseptic surface because it looks clean. And this could not be further from the truth. And here is a slide published through the American Society of Microbiology. It shows listeria, which had been contaminated onto a stainless steel surface. The surface was then cleaned as best as the laboratory staff could do so, and then they looked under the electron microscope. You will see within the scratch marks on the stainless steel that we have bacteria embedded in there. Now, this is a surface cleaned as well as possible. What it shows is it's very, very difficult to clean a surface. So unless you've got good cleaners and unless you've also incorporate disinfectants into those cleaners, this is what you're leaving behind. And if you don't keep up with regular cleaning, if that surface becomes contaminated with, for example, skin cells or blood or what have you, then there's a risk that they will continue growing on that surface, form biofilms, and increase the risk of cross-contamination and spread within that environment. So people really have got to wake up to what stainless steel is and what it actually does. It may be corrosion resistant, but the emphasis is on the cleaners, the workers, to keep it clean. Stainless steel itself does not have intrinsic antimicrobial properties. Now, people have used a Japanese standard test, the GIS test, and I have to say that myself and my colleagues have looked at this test. We're very disappointed with it. It's a test where they incubate pathogens under a film of water and a plastic film at 37 degrees C. It's not really representative of the built environment at all, as far as we can see. And in our studies, I'll show you some in a minute, we get very good survival of pathogens on stainless steel. So I think we have to be very careful about how we interpret international standards and how they are applied to show, to show how they are applied to the assessment of efficacy of antimicrobial surfaces. Now, in our work, we've looked at a variety of surfaces. For the MRSA studies, I'll just show you some of the alloys we've looked at, including pure copper, brass, nickel silver, copper nickel, tin bronze, and also a more novel alloy, Y90. So in these experiments, we've taken MRSA, and this is at room temperature, 20 degrees C. It's at a normal relative humidity, which for the UK is about 50% to 60%, although we've also done this work at 0 humidity and 100% humidity, and you get exactly the same results. We put the organisms onto stainless steel, and we inoculate, in this case, at 10 million cells per centimetre squared. Why such a high inoculum? Well, this is gross soiling, but also international standards ideally would like people to show a log 6 reduction. This is approved within the food industry, and it's something that the healthcare industry are also looking for. So we start off with very high concentration inocular. You'll see here that over a period of 6 hours, we get hardly any decline in the numbers on stainless steel. Now, in these experiments, we do replicates of at least 6, frequently repeated over 3 to 4 occasions. So we're looking at many data points in here, and the data are very tight. Now, by comparison, when we incubate on pure copper, you'll notice here that we're getting a complete kill, so that's over a log 7 kill, in under 1 hour. And then when we look at the brass alloy, which is 80% copper, 20% zinc, we also get a kill, although this takes a little longer, but around about 4 hours to get a complete log 7 kill. With the alloys with a lower concentration of copper, we also get a kill, but this takes longer, 6 hours or so. But you'll notice on the stainless steel, it's just surviving for a long time. Now, at those 20-degree experiments, we've actually run them out for weeks, and in fact, the MRSA is surviving very well on stainless steel for 6 weeks, that we've seen for sure. Now, we also were interested in what would be the effect of repeating the experiments at 4 degrees centigrade. Several reasons for this. If you're in the food industry, 4 degrees is very important because of food processing. But it occurred to us as well that in the hospital environment, if you think of the mortuary, when cadavers are chilled, they're being held at a chill temperature. So again, if you're concerned about cross-contamination in the mortuary, looking at 4 degrees centigrade is an important temperature to look at. Again, this is the stainless steel data. We get very good survival over a long time period. This is the pure copper. Again, we're getting a complete kill, log 7 kill, but you'll notice now it's taking us about 3 hours. Now, this is not unexpected because microorganisms follow the laws of physics and chemistry, and when you lower temperature, you lower chemical reaction rates. So all that happens is it takes longer to kill them, but you still kill them. And then when we look at the brass, for example, again, if we run this experiment on, we'll get a complete kill down here after 6 or 7 hours, and some of the other alloys also. So again, even at 4 degrees C, we're getting quite a good effect. Now, as a microbiologist, I have to warn you of something, and that is we have relied on culture techniques for over 100 years. They've stood us in very good stead, and they tell us a lot about whether water is safe, whether food is safe, whether patients are safe. But as our knowledge of microbial physiology is improving, we're starting to realize that there's a new state for bacteria, and it's what we call sublethally stressed cells. Some people call them viable but non-culturable cells. And there's growing evidence that when you stress some pathogens, they go into this, if you like, semi-dormant state. What that means is they may still be alive, but you can't culture them. So it's no longer sufficient just by using culture techniques to say you've killed an agent. The onus now is to go on and use new tests to really confirm that. So one of the things we've been doing is using this metabolic stain, which is called CTC. It's got quite a long name. What it is, it's a fluorescent redox dye, and it picks up the respiration of microbial cells. So if they're alive and respiring, they pass electrons to CTC. It becomes reduced, and when it becomes reduced, it fluoresces red. So we have a very simple test to show if an organism is respiring or not. So here's looking at those surfaces under the microscope. There's stainless steel, there's copper. Now what you can't see very well on here are all the cells. What I can see on my computer is in this picture here, this is MRSA on stainless steel. They've been on the stainless steel for 3 days, and according to our microbiology test, they can be cultured, and therefore they should be respiring. If they're respiring, they should make CTC go red. And what you actually see are a lot of red stained cells, so they're alive and respiring. If you look on copper, there's absolutely nothing. The bacteria are there, but they're not respiring. So the copper has killed their respiration, and that collapses the membrane potential. These cells are dead. Now, one of the questions that we were asked when we submitted one of our papers was, how do you know with the alloys that it's actually copper that kills the cells and not one of the other metals in the alloy? So we did some pure culture studies using single metals. And in this case, you can see the data for nickel. And again, doing the experiments at 10 million cells or so, you'll notice that over a 6-hour period, we get hardly any decrease in the numbers. So nickel is not antimicrobial over this time course. Equally, when we look at pure zinc, again, you see we get only a very slow decrease of the cells on zinc. And if you remember on copper, we're down here. So it looks as if the antimicrobial effect is truly due to copper and not due to these other components in some of the alloys. Now, of course, one of the interesting things about the copper industry is that the metallurgists are coming up with new alloys all the time. And these alloys are being developed for a whole range of different uses. Are they corrosion resistant? Are they tarnish resistant? Can they be used in high shear and wear engineering applications? What we don't know is whether they also have intrinsic antimicrobial properties. So we've started looking at some of these. So here, for example, is this alloy 706. And you'll see it's 87% copper, and it's also got zinc, nickel, iron, and manganese in small amounts. And again, you'll see that from the kill curve, it's almost identical to pure copper. So even these new generation of alloys, although they contain less than 100% copper, are giving us very fast, very efficient kill of MRSA, which I think is very exciting for the future, because I think it shows it should encourage metallurgists to go on and look for new combinations of alloys, hopefully with the same antimicrobial properties of pure copper. And I think this will open up a new era for the industry because of that. Here's some data looking at C510 tin bronze, which is 95% copper and 5% tin. And again, we get this very impressive kill curve in under an hour, coming from 10 million cells to start with. So a lot of those studies were actually looking at high concentration inocular. What we really wanted to know is what's going on in the real world. And until the recent studies funded by the CDA, there's been very little data in the literature on quantitative assessments of pathogen load in hospital environments. There was one study published in 2002, which was a hospital in London. And when they sampled surfaces within the hospital, they found typically that on a door handle, you got between 1,000 and 6,000 MRSA per door handle. Frequently, it was round about 100 colonies per centimetre squared. So we said, well, we'll really better go back and repeat our studies and look at these truer concentrations. So we repeated the experiments, but now we actually reduce the concentration going onto the surfaces. And in these studies, this is a repeat of the original 10 million work on copper. And again, you'll see that we're getting this impressive kill within 1 hour. But you'll notice that as we reduce the inoculum, the load of cells on the surface, the kill time is increasing even more. So in fact, if we have a loading similar to what was reported in London, we've got a complete kill in 15 minutes, which I think is very impressive indeed. So what it's forced us to do now is reevaluate the way we test all of our surfaces. We still do the high inoculum dose challenge to show log 6 or log 7 kill, but we're also looking at more realistic concentrations, typical of what you might find in the hospital environment. And I'm really looking forward to Mike Schmidt, Tom Elliott, and others publishing their data so that we've got good references to be able to relate to and say, well, these are the concentrations we actually find in the hospital environment so that we can set our experiments accordingly. Now, there isn't a lot of time to discuss this, but what is copper doing? Well, there have been a whole range of potential mechanisms that have been published. So, for example, how does copper disrupt cell function? Does it inactivate proteins in cells by binding to them directly? Does it inactivate RNA or DNA by binding to them directly? Does it disrupt enzyme structure? There's also a school of thought that copper works indirectly. Because copper can cycle through copper 0, copper 1, and copper 2, this redox cycling can be involved in reactive oxygen species generation. And there's several groups who actually believe that there's peroxide complexes, or in some cases hypochlorite complexes being formed, and these are actually doing the killing effect. Whatever, I think a lot more work needs to be done in that area. But what I will do is I'll show you some of the sites of action. So, just to remind you, this is the respiration experiments where we have MRSA on stainless steel, and you can see the cells are respiring, and therefore they're red. And this is on the copper, after only a few minutes, and they're not respiring. They're all dead. Now, there's another set of stains we can use, and it relies on the fact that we have a small green fluorescent stain. And this stain is so small, it goes into all cells, whether they're alive or dead. It goes into the cells and it stains their DNA, and so the cells look green. Now, by contrast, there's another stain, propylium iodide, and this is a very large red fluorescent molecule. It's so large, it can only go into cells with compromised membranes, i.e. dead cells. So, if it goes in, they stain red. So it's a simple assay, green is alive, red is dead. And so you can do this work, and what we've found using this stain is that for MRSA, the majority of cells stain green, but only a few of them, I don't know how well you'll see this on the projector, stain red. So in fact, what it's telling us is that many of the cells have actually retained an intact cell membrane. Now you might feel, well, big deal, so what? Well, it's important because last year a paper was published by Elliott and Varan. And Elliott and Varan suggested that MRSA will survive on copper. Ignoring the work that we'd published, ignoring the work that CDA had funded through the GLP lab in the USA, ignoring the work of Shaheen Mehta, who'd also published last year, repeating our MRSA work. And the reason that they suggested that MRSA survived was because they used this stain system. And they said, oh, look, none of them have stained red. Therefore, they must be alive. But there's a fundamental flaw because you've already seen our other data. These cells cannot be cultured. We've killed their respiration, even though they don't take up this stain. So what that means is that last year's published paper is an artifact and actually should be ignored. Unfortunately, some people are reading that and they're questioning whether that compromises the copper work. So we've just submitted a paper to a journal where we're trying to rebut those original studies and say, sorry, they got it wrong. These organisms are dead on copper. Now, there's been some interest in whether copper actually degrades DNA. So in these experiments, we took MRSA and we put them onto stainless steel, and then we recovered the cells and we extracted the DNA. And you can see a band here relating to DNA. When we did the same experiment on copper and extracted the DNA, we've lost the band. And then we've just got some very, very low molecular weight material down here. So that's suggesting that copper is actually destroying DNA. And there wasn't time to actually put it in my presentation today, but we've developed a new assay where we can look at total genomic DNA under a microscope, and we can confirm these experiments. So copper is killing the respiration of cells, and it's totally destroying the DNA. The DNA is gone. Now that's very important because people are saying, what is the risk that copper will form mutations in bacteria? And through those mutations, the bacteria will either become resistant to copper or acquire resistance to antibiotics in the same way that triclosan has been implicated in antibiotic resistance emergence in pathogens. Well, this work tells us the DNA is gone. We've wiped it out completely. These bugs have lost their DNA, they're dead, there's no chance of mutation at all. So I don't consider that to be a problem. Now, if we move on now at Enterococcus, vancomycin-resistant Enterococcus, which several speakers have already said is an emerging problem around the world. Here's some work with Enterococcus faecium. And again, you'll see that, unfortunately, again, Greece doesn't come out very well in this, but equally, neither does Portugal or Ireland. It's an increasing problem, and more of the isolates we're picking up are vancomycin resistant. So this is one of these emerging infections, if you like, a new superbug. Again, there's very little published work so far. We found one study last year by Eckstein, and they looked around a hospital environment, and they were able to isolate the vancomycin resistant enterococci, VRE. They found it on a whole range of hospital surfaces, including bed rails, telephones, call buttons, doorknobs, toilet seats, and bedside tables. So it seems to get around as much as MRSA does. We repeated the work, looking at survival on the copper alloys. And here's some work. I'm sorry this hasn't come out very well since I sent the presentation by email. But to summarize that on pure copper, again from this high inoculum concentration, we're getting a complete kill in under 100 minutes. And even the other alloys which we've looked at, such as brass and copper nickel, are also giving us a very impressive kill very quickly indeed. So it looks like VRE is responding in a similar way. So we then went to do the low inoculum experiments, and again, when we cut the concentration down, this is actually looking on stainless steel, and at the high concentrations, we get very good survival. We've actually run this experiment out now for a couple of months. I mentioned this sublethal stress state, viable, non-culturable. You might conclude from this experiment that all these cells were dead, but we ran the experiment on, and you'll see they've actually recovered. They've gone through this physiological adaptation, and they've actually recovered. So you have to be very careful when you do your microbiological analysis to convince yourself what is alive and what is dead. Only at the lowest concentrations did we seem to be losing the culturability. But look, it's taken us 20 or 30 hours to actually get these low numbers, where on copper we've done that in minutes. So it's a big difference. We've looked at a whole range of strains. In this case, this is Efficacy, which is vancomycin resistant on pure copper. Again, we're getting these very rapid kill curves. At this one, which is the concentration which we guess is going on in the hospital environment, we're getting a kill in 20 minutes. So very, very impressive. And we can repeat all of that work looking at a range of alloys. There isn't time to go into great detail, but you'll just see from the trends of the kill curves that on all the alloys, particularly as we lower the concentration to those we might expect in a hospital environment, we're getting complete kill in just a few minutes. Okay. Again, we wanted to know, could we confirm this loss of culture in terms of cell death? So in this case, what we've done, we've repeated work with the green stain. So this is the green stain here to show the bacteria there. But now, rather than use that red stain, which was the big red one for membrane integrity, we now know actually that, again with VRE, the membrane is not compromised. So what we've done is we've gone over to this other red stain, the CTC, which measures the respiration. So what we can now do is say, here's stainless steel, here's the cells, and you can see them respiring on the stainless steel. And down here, we've got the two species, E. faecalis and E. faecium, and again, on the stainless steel, they're respiring. When we go over to copper, you're struggling to see anything. Now, it's telling us two things. The first thing it's telling us is that they're not respiring. They're not CTC red, okay? But the other thing, which initially surprised us, but then we thought about it and it's not really surprising, this DNA stain, it's not detecting DNA. Now, I've already shown you with MRSA, we've destroyed the DNA when they're in contact with copper. So really, this shouldn't surprise us. The DNA is gone, so therefore the Cyto9 cannot stay in the cells because they've lost their DNA. Moving on quickly now to Acinetobacter, you've already heard about what a problem this is starting to be. It causes very serious wound infections, which are very difficult to treat, and again, amputation is frequently the outcome. It's a major concern to clinicians. In the UK and the US, we've had major problems with troops returning from the Gulf. The troops are actually bringing back the Acinetobacter baumannii, and they're then transferring them to patients in hospitals. So, for example, in Tom Elliott's hospital at Selliok in 2003, 2 troops returned from the Gulf. 93 people were infected, and 35 died. So this is really a serious problem, and the military are just as concerned as the normal civilian officials as well. So we had to try what happened on copper. We started with very high concentrations, and you'll notice here that we're getting a complete kill from this high concentration in 3 hours or so. The alloys, such as brass, are taking longer, 24 hours, but we're still getting a complete kill. Now, that may not look as impressive as MRSA, but this is a really tough beast. People really struggle to control this and to kill it using normal disinfectants. So that's pretty good. So we then repeated our experiments with the low inocular concentrations. And you'll notice here that when we come down from these very high concentrations, so we actually went up to billions of cells at one stage and got complete kill in 6 hours. When we come down to thousands of cells, we're getting complete kill in 1 hour or less. So it looks again within the hospital environment as if we're going to get pretty good control of this pathogen using copper. Again, we can use that respiration experiment And again, I don't know how well you'll be able to see this, but on stainless steel, you can see red respiring cells at 0 hours, 5 hours, and 24 hours. Whereas on copper, we start off with some respiring cells, and very quickly, they lose their respiration. So certainly between 3 hours and 5 hours, they've stopped respiring, membrane potential has collapsed, and they're dead. We've repeated this DNA assay, and again, they've lost their DNA. It's gone. And then membrane integrity, there isn't a lot of time to discuss this, but just to show, again, on copper, some of the cells are picking up the red stain, showing that the cell membrane is compromised, but some of them are actually not taking up the stain, showing that, in fact, the membrane's intact. If we look at Clostridium difficile, which you've started to hear about now, it forms these spores which are highly resistant to cleaning and to disinfection. This is a major challenge for the hospital environment. When we look at these, if we look at a combination of vegetative cells and spores, then we can get a good kill within 24 hours on copper and longer kill on brass, but it still is achieved. And when we look at just pure spore preparations, again, we can get a very good kill from 1 million cells down to 0 in 24 hours, 48 hours for the brass. Now again, that may not sound very good, but we're dealing with spores. Copper is killing the vegetative cells in minutes. It's got to work harder to kill the spores, but it gets there in the end. And again, we can use the CTC assay to actually look at the metabolism of the cells. And what you notice on the alloys after 24 hours, well, actually you won't notice it because there's no red cells here, whereas on stainless steel, on my computer, there's actually a lot of red respiring cells. Okay. And then that's just some more work backing that up. So we can conclude from all the bacterial studies that copper alloys are doing a good job. They're intrinsically antimicrobial. And this work has been supported by other laboratories who have shown similar effects. Now, just very briefly, we've gone on to look at fungi, the Aspergilli in particular, because they're well-known causes of opportunistic infections, allergic states, and toxicoses, particularly in hospital environments where you have immunocompromised or asthmatic patients. Again, we can do those experiments using from high inocular concentration. If we look at 3 species, Flavus, Niger, and Fumigatus, if we look on copper, then we see that Fumigatus and Flavus are killed on copper within 96 to 130 hours, which again is not much, but we're looking at spores here. On stainless steel, they just run forever, weeks and weeks and weeks. In the case of Niger, we got a strange result. The Niger appeared to survive. It didn't look as if the copper was killing Aspergillus niger. But we then went on to show that, in fact, if you put them onto copper and stainless steel and see if they grow, the Niger would actually grow on stainless steel. You can see the hyphae, which come from the outgrowth of the spores. And if you look on copper, all you see are the spores. There's no outgrowth. What that tells you is that copper is sporostatic, not sporocidal. And again, you can confirm this. Here's stainless steel coupon on a spore growth medium, and there's copper. There's no growth of the spores and the hyphae onto the copper, but we're getting engulfment of the aluminium surface with the growing hyphae. So when you come to think about high-velocity air conditioning systems, it looks like copper is going to be a superior material compared to aluminium when we construct the systems, the cooling ducts, what have you. Very briefly, influenza. Influenza kills about 12,000 people a year in the UK, 36,000 in the USA, so it's a major killer. It's well known that it survives on surfaces, it's easily transferred through contaminated hands, and in fact, one set of contaminated hands can contaminate 7 other sets of surfaces or hands. We developed an assay system where we looked at infection and survival. It was a cell culture-based system where we could titrate the number of infectious particles into these cells, and then green cells actually show infection. So that's what happens when you have 10 million flu virus in there. This is what happens when you have 10,000, 100, or what have you. So using that calibrated system, we looked at survival on stainless steel. And this is what happens. So we started off with 2 million inoculum on the stainless steel. After 6 hours, we've still got 1 million infectious virus surviving. After 24 hours, we've still got 500,000 infectious virus surviving on stainless steel. When we look at copper, within 1 hour, we've only got 500,000 infectious virus surviving. 6 hours, they're virtually all disappeared. So we've got a 1 million kill or greater in that time. Adenovirus, this is a virus associated with respiratory and fecal infection. It produces severe coughing, high fevers, damages the conjunctiva, causes meningitis and encephalitis. So again, it's something else we're very concerned about, and it survives well on surfaces and again is easily transferred between hands. So again, using the assay similar to what we developed for flu virus, then we can look, for example, on stainless steel, and there's what happens after 6 hours contact on stainless steel. Most of the inoculum is still alive, and there's 24 hours contact on stainless steel. Again, a lot of survivors. By contrast, on copper, within 6 hours, we're struggling to find anything, and certainly nothing within 24 hours. So again, copper is giving at least a million times kill within a few hours. So my final concluding slide to all of this is that I hope I've been able to show you in the short time that copper is strongly antimicrobial against bacteria, viruses, and fungi. It certainly outcompetes rival materials such as stainless steel and plastics. And we think that it's going to be really promising, particularly with this new generation of copper alloys coming out, where we can build antimicrobial properties into their other resilience properties. And I think because of that is that I personally see a bright future for copper and the alloys. Okay, thanks.